Is it lonely over here? Yeah. I was thinking about like getting some fun costumes for the skeletons and things around here so I can have like a fake co-host or something. But yeah, it's lonely. Lonely life out here. I'm ready to start. Are y'all ready to go? Got 16 people here. That's something. 17. Yay. Here I know. What should I name this one? Yeah, there are professors here. They're working on this too. I'm just grateful I know how to use these programs pretty well. Some There's a steeper learning curve for others. Henry or Henri? Dr. Phil? No, get out of here. I'm more inclined to choose a name that's a pun. So if you can think of a pun, then I'm down for that. What can we do? I should turn some lights on. Okay, what we're going to do first is I'm going to go over some models and some histology based on what we just discussed. Dry bones. It's not very clever. Oh, Mario Kart? Is his name Dry Bones? Okay. So this is a model of a neuron. This is a model of a neuron. So it's a weird looking model. It's only a part of a neuron because you've got the cell body over here, you've got an axon coming across, and then it's cut off. So there should be more, of course. Verbenjager Manjensen. Who's that? All right, let's take a look at this. I haven't thought through how I'm going to show this. That works, I guess. Okay. Okay. This is the cell body. This part is clear just so we can see what's on the inside, but it's supposed to be continuous with this more opaque looking thing. So this is the plasma membrane of the cell body. This is the plasma membrane of the cell body. These harder protrusions here, whether it's clear or not, I'm talking about the hard one, like this, those are dendrites coming off of the cell body. So this is where the receptors would be on the dendrites. You can have receptors on the cell body as well. These things, these weird rubbery Hershey Kisses looking things, those are actually axon terminals of different neurons. So this does not belong to this neuron. This belongs to another neuron that's communicating to it. So this is a synapse, this is a synapse, axon terminal synapse of the dendrite. This is receiving lots of input. It's got a lot of dendrites, it's receiving lots of input. So who can tell me what shape of neuron this model is? What is the shape of this neuron? How long is this lag? Let's see. No answers yet. Got lots of dendrites, lots of input. It's multipolar. That's right, Emilio. It's multipolar. So we have a multipolar neuron here. You can see the nucleus and all the other organelles on the inside. You can see the axon hillock right here. So if the axon hillock is where we have lots of voltage-gated channels, this is our decision point. Are we going to have an action potential or not? See how the axon continues along here. We've cut into the axon so we can see inside of the axon. We've got stuff surrounding the axon. You can see a nucleus here, and then all these layers. So this is myelin. So this is a myelin sheath. This is a gap. This is the node of Ranvier. Myelin sheath, another gap. So if this is a single cell wrapping around the axon here, and then here's another single cell wrapping around the axon, what kind of cell is this? Which glial cell is wrapping around the axon? Go ahead and type it in the chat. That's right, Schwann cells. These are Schwann cells because these are individual cells wrapping around. If they were separate, if it was an oligodendrocyte, the cell body would be out here and it would send out a projection that would then wrap around. But these are individual cells wrapping around with gaps in between. Given that these are Schwann cells and that given this is multipolar, can anyone tell me what kind of neuron is this? Is this a sensory neuron, a somatosensory neuron, a special sensory neuron, an interneuron, or a motor neuron? Those are your four choices. Special sensory neuron, somatosensory neuron, interneuron, motor neuron. Given that it's multipolar and has Schwann cells, what kind of neuron is this? The choices again are special sensory, somatosensory, inter, and motor. That's right. This is a motor neuron. So that means the cell body and dendrites, this would be somewhere in the central nervous system, whether it's the brain or spinal cord. This axon extends out into the periphery, which is why you see Schwann cells. So this is representing a multipolar motor neuron. I just want to reiterate, you can look at this cell from a longitudinal cut. You can look at it from a cross-section cut. You can see, oh gosh, you can see the axon in the middle. You can see the myelin wrapping around. This sheath covering the outside, that's covering the myelin, this is called Endo Nerium. Remember we went over endomysium, which covers up individual muscle fibers? This is Endo Nerium. I'll type that out. Endo Nerium. Endo Nerium is a protective connective tissue that surrounds axons. So that is a model for a neuron, specifically a motor neuron. I am going to continue the notes that we were just on because they're relevant to things I'm going to show you here in lab. So I'm going to switch over to the slides. We left off on slide 30. This is slide 30. Oh, God. Oh, poor Henri. Are you okay? I'm okay. Okay. He's fine. Yeah? Okay. You're mad at me? Okay. So, was the pink thing on the cross section the nucleus of the Schwann cell? It was. It was. Well, so here's the axon. So this is not a nucleus. This is the axon cross section. Then you have myelin, and then here's the nucleus of the Schwann cell. Nucleus of the Schwann cell. Does anyone watch Bob's Burgers? I want to be like Bob when he talks to his food or whatever it is he's talking to. Let's give him that high-pitched voice. Okay. When we look at nervous tissue, brain or spinal cord, we can divide it up into gray matter and white matter. Gray matter is grayer. I mean, it's not perfectly gray, and white matter is not perfectly white either. But gray matter appears darker because this is where you find a cluster of cell bodies. Gray matter is where you find mostly cell bodies. Here's a frontal section of the brain. Oh, I had a good one. Hold on. Where is it? Ha. So we're taking a frontal section of the brain. So brain, frontal section. You can see that the darker gray is towards the outside. That's because this is where you find cell bodies. The gray matter is where you find cell bodies. So you can see that here. And when we're looking at the brain, it's towards the outside. When we're looking at the spinal cord, though, when we're looking at the spinal cord, it's towards the inside. So the gray matter forms these wings. It looks like a butterfly. So gray matter is towards the inside on the spinal cord. Gray matter is towards the outside on the brain. The term for outside of an organ, that's cortex. So you find gray matter in the cortex. I know you might want to say core, it's towards the middle. Cortex is towards the outside. And the cortex of the brain is where you find gray matter. The term for these butterfly-shaped things in the spinal cord where you find gray matter, these are called horns. So you find gray matter in the horns of the spinal cord. You find gray matter in the cortex of the brain. If you have clusters of gray matter that are deeper in the brain, so you can see that there's gray matter on the outside, but then you also have some gray matter towards the inside. If you have clusters of gray matter, clusters of cell bodies somewhere in the middle of the brain, deeper in the brain, not on the surface, that's called a center or a nucleus, or plural centers and nuclei. All that term means is a centralized cluster of something. We've used this term to describe the thing where DNA is inside of cells. And I sure I've used it for something else, but that's all that means. So you've got a cluster of neurons deeper in the brain, just not on the outside. The outside is cortex. If you have a cluster of gray matter on the inside, that's a nucleus or a center. If you have a cluster of cell bodies that's outside of the central nervous system, a cluster of cell bodies in the peripheral nervous system, those are called ganglia, singular ganglion, L-I-O-N instead of L-I-A. Ganglia, so back to our spinal cord. Here's the spinal cord. Here's the posterior dorsal side vertebrae, spinous process. Over here, this is an example of a ganglion. In this area, we've got cell bodies that extend to the spinal cord. Remember how I showed you all this sensory neuron has its cell body off to the side and then its axon goes into the spinal cord? This cell body is within a ganglion. Because in this area, you're going to find more cell bodies. There's a cell body here, a cell body here, a cell body here, for other neurons. You have a cluster of cell bodies in a ganglion outside of the spinal cord, outside of the different types of gray matter. We've got cortex, we've got horns in the spinal cord, we've got ganglia, and centers and nuclei. Questions? Okay. I'll keep looking out for the chat. Let me know. Okay, so the opposite of gray matter, where you don't have gray matter, you have white matter. Gray matter appears darker because there are more cell bodies, more nuclei. White matter appears lighter because this is where you have myelin. This is where you have axons. Remember, myelin is made of fat, lipids. Lipids is a lighter color. Lipids are a lighter color. If you ever see the fat on a steak, it's lighter. Right, back to our frontal coronal cut of a brain. You can see how it's lighter towards the middle. These are all axons going to and from neurons in the cortex and the nuclei back and forth. This white matter, these are all axons. Some axons go cross hemispheres, some go down the spinal cord, some just cross from cell to cell in the same area. That's white matter in the brain. White matter in the spinal cord is towards the outsides. They're called columns. So here's a column of white matter, here's a column, here's a column. Think of this as a cross section. So the axons are coming out at you. They're going up and down the spinal cord. The axons are. So they're found in tracts in the brain. They're called tracts. If it's in the spinal cord, they're called columns because you're going in a column. Tracts and columns. They appear lighter because this is where we have axons. There are some cell bodies, but very, very few. There are cell bodies of glial cells typically. You don't find neuronal cell bodies in white matter. No neuronal cell bodies in white matter, only in gray matter. When you take a cross section, oh, I wanted to show you this. So here's an actual brain that's been cut. Trying to reduce the reflection here. You can kind of see it. Okay, so here you can see these spaces. Those are ventricles. That's where cerebrospinal fluid would fill up. You can see there's lighter color here. The lighter color, that's white matter. On the outside, it's darker. That's gray matter. There's actually a tumor here, but we'll get to that later. Or a, what do you call it? Brain damage. Probably a stroke or something. Anyways, you can see white matter, gray matter. All right. Nerves, what is a nerve? A nerve is, it's an extension coming off of the brain or spinal cord. So where can I show you a nerve? You're gonna come with me. All right, so we're looking at our friend here, Mr. Torso, and you can see that the spinal cord would be, in the back here, but we can see an extension that would be originally coming off of the spinal cord. We can't see because it's hidden, but what's labeled here in yellowish, this is representing a nerve extending out. So this is a nerve coming off of a spinal cord. Or where's another good example? Where are those vertebrae? Right. So here is a spinal cord, remember? Pelvis, spinal cord, vertebral column. These are nerves extending off of the spinal cord. The nerves are exiting or entering from these intervertebral foramina. Those are intervertebral foramina, and you've got the nerves coming out. So all a nerve is, when you say nerve, nerve is not a neuron. Nerve and neuron are two different things. A nerve is a collection of axons. A nerve is a collection of axons. You've bundled them together. If anyone is a fan of cable organization, it's like that. You're bundling all those cables together so that they're more organized. So spinal nerves, nerves coming off of the spinal cord, they're found in the peripheral nervous system. By definition, it's outside of the spinal cord, outside the brain, it's peripheral. They are protected and organized by connective tissue. And guess what? It has that same pattern that we saw with muscles. Represented here is an axon and myelin sheath. You can see the nodes. Surrounding a single axon and myelin sheath is Endo Nerium. Endo Nerium surrounds a single axon / myelin sheath. It's Endo Nerium. Perinerium is when we have a bundle of those axons surrounded by a sheath. Perinerium is surrounding a fascicle. We've seen that term before. All fascicle means is a bundle of something. We're talking about a bundle of axons. So perinerium surrounds a bundle of axons. On this histology here, this cross section of a nerve, it's very, very tiny, and I'll show you a better picture coming up. You can see those tiny dots, that's the axon. The circle is a myelin sheath. And so surrounding that, we can assume is Endo Nerium. You can see that it's been bundled. So this is one bundle of axons. So this is where you find perinerium. When you bundle together all the fascicles, then you want to cover it with connective tissue all the way around. That's epinerium. Epinerium is the outermost layer of a nerve. It's continuous with dura mater. We'll get to that later. It's the protective covering of the brain and spinal cord. But the whole purpose of this is to organize everything, just like what we did with muscle fibers. We want to make sure everything's protected. We have 3 layers of connective tissues. Oh, let me show you that on our actual histology. Peripheral nerve 59. Okay, I want to show you one example here. How well can you see this? Not that well. Hold on. Any better? Okay, we are looking at 3 nerves here that have been cut. So here's one nerve, really big. Here's another. So let's focus on this one. You can see that this is the whole nerve. We're at what are we at? We're at 40x right now. So this is the epinerium. Surrounding the whole nerve, this is epinerium. Let me zoom in. We're going to 100x. We're at 100x. This is still epinerium. Perinerium, let me show you a different part here. The boundaries of this fascicle aren't the most visible here. So I'm going to do my best to point it out. You can see a line here. This line here. It's a bit darker, extending from this darker red part. Goes around, and then it continues up this way. This is a fascicle. I know I can't see the whole border like over here, but this is a fascicle. So this darker red, that's perinerium. Endo Nerium is surrounding an individual axon. So you can see the axon here in this darker pink. The white is the myelin, and then Endo Nerium is forming the circle around that. So Endo Nerium, perinerium, epinerium. This is one slide of a cross section of a peripheral nerve. Let me show you the same thing, just a different stain. This one shows it very differently, and you can see the fascicles a bit better. So we're at 40x right now. We're at 40x right now, and already you can see a big difference. You can see the outline of the whole thing, and you can see individual fascicles. Let me go to 100x. So this is the outside. This is epinerium. A fascicle would be right here. So this is perinerium. Oh, there we go. Perinerium. And then here, this little dot, these little dots that you see here, those are axons, myelin sheath, Endo Nerium. Okay. You have any questions? Go ahead and type it in the chat. Let me know. We've talked about a bunch of different receptors, so just to recap and to add a few more, for somatosensory receptors, Merkel cells are special receptor cells. So what that means is this is your epidermis, your stratum basale. And so here you have a Merkel cell. A Merkel cell is its own cell. It communicates with a sensory neuron that's found just deep. And that sensory neuron goes somewhere else. It's a pseudo unipolar neuron. So a Merkel cell is a special receptor cell. Something like a Meissner's corpuscle. You still have a nerve ending, a dendritic ending like this, but you've got connective tissue surrounding the dendrites. That's Meissner's and Pacinian. Yeah, Merkel. So Merkel cells, stratum basale, light pressure and texture. Meissner's corpuscles and the dermal papillae, flutter and stroking. They're also fairly superficial, pretty light stuff. We've talked about those two, and we've also talked about the hair root plexus. It's the nerve endings, dendrites. They're bare. There's no connective tissue. So the term for that is free nerve ending. Bare dendrites that are wrapping around the hair, and so that senses hair movement. So those are three that we've gone over. Oh, we've also gone over Pacinian corpuscles, to deeper connective tissue covering that senses vibration. Two more, I guess 2.5 more that I want you to know. Ruffini corpuscles are also deeper in the skin, connective tissue covering. They sense stretch, skin stretch. And then to detect pain, the word for pain is nociception, the reception of pain, nociception. And temperature, thermoreception. Both of these are free nerve endings, meaning, where are we? Like this one here. It's a free nerve ending. It's bare dendrites to detect some kind of noxious stimuli that causes pain or temperature changes, like how fast molecules move. Those are all examples of somatosensory receptors. Who can tell me what is the shape of these sensory neurons? What is the shape of these sensory neurons? What do I mean by skin stretch? Like literally stretching your skin. Pulling on it. Yeah, these are all of these neurons, all these sensory neurons are pseudo unipolar. If it's somatosensation, there we go. Somatosensation, it's pseudo unipolar. Same with a muscle spindle. Same with a muscle spindle. Muscle spindles are the ends of neurons that are pseudo unipolar. The term for detecting muscle stretch and skin stretch are two different things. The term for detecting muscle stretch, or for how stretched your tendons are, or your body position, that's called proprioception. Proprioception. So you need proprioceptors to detect those things. Body position, tension, muscle length. The whole purpose of these receptors are to make sure your muscles don't get damaged. Like the reason why you kick your leg out when you tap your tendon like that is because you want to make sure that you don't overstretch your tendon. So it's a protective reflex to kick your leg out. You're kicking your leg out so you don't overstretch your quadriceps muscles, like your rectus femoris. That's what a stretch reflex is. A stretch reflex is to prevent overstretch, and that's mediated because you have muscle spindles. Muscle spindles monitor how stretched out those muscles are. Under a microscope, this is a cross section This is a cross-section of a muscle spindle. You can see regular muscle fibers, cross-sections of muscle fibers out here. You can see more muscle fibers inside of this capsule, but that's the difference. It's covered by this special connective tissue capsule. A muscle spindle does have muscle fibers surrounded by connective tissue, and what you can't see are nerve endings, dendritic things within here. For reasons that we're not getting into too much right now, you'll hopefully learn in physiology. This whole thing, this muscle spindle, this is the thing that detects, and you need specialized muscle fibers inside for allowing for that, for them to protect things like with the stretch reflex, to detect the stretch. So this is for detection. I know it has muscle fibers in here, but they're not for contracting. They're for detecting. These are special muscle fibers inside of muscle spindles for detecting stretch. We do have a slide of this, so let me pull that up. Okay, all this should look pretty familiar, hopefully. We are at 100x total right now. We've got skeletal muscle in longitudinal section. We've got an axon and axon terminals. So these are axons of somatic motor neurons to control everything going on here. But here we can see a branch of something coming off of this bundle, and then coming in between these fibers, and it's much thinner. This is a muscle spindle. This is a muscle spindle. So you can look for the nerve endings. So these are dendrites, not axons, coming off of the spindle. These dendrites and coming in between the larger muscle fibers, this is a muscle spindle for detecting muscle stretch. Again, this is 100x. There is another receptor type for proprioception that we don't have the histology for, unfortunately, but I can show you what we have here. There we go. Another receptor you can find in muscles is called a Golgi tendon organ. It's that guy's name again, Golgi, but it has nothing to do with Golgi organelles. That's different. Golgi tendon organs, they also detect how stretched out your muscles are, but really not how stretched your muscle is, how stretched your tendon is. If you're pulling on a muscle, if I'm stretching my muscle, if you're stretching like the meat of the muscle, it's not the end of the world. But if you're starting to stretch the tendon, that's really bad. If the tendon is getting stretched, that means you're about to pull it off the bone. That's really bad. That means your tendon's getting overstretched. You need this organ, this receptor, I should call it. You need the Golgi tendon organ, a receptor, to detect overstretch. With the muscle spindle, the whole point of the reflex was to kick things out, to shorten the muscle again. This is to completely inhibit or relax your muscle. So imagine like you're trying to carry something heavy and you're like struggling, and then someone puts on even more weight, and if you were to keep holding, your muscles would tear. So you just reflexively let go. That letting go, that's a reflex initiated by your Golgi tendon organ, detecting that excessive stretch. How does that look in histology? Again, we don't have slides of it, but I can show you the histology that we have here. You can see the muscle fibers here. They come to a point, and here's our Golgi tendon organ, dendrites leading to an axon. As the name implies, it's going to be found in tendons, while muscle spindles are found within muscles. Those are Golgi tendon organs. So I hope that all made sense. We've gone over how a reflex can even happen. We have to sense something. We send that information to the central nervous system, whether it's a monosynaptic or polysynaptic reflex, and then send that information outward to somewhere else. Could be the same thing, could be somewhere completely different. Just depends. Take a moment, please. Okay. Looks like most of you are saying C. If you leave the fascicles and axons together and intact, that means the Endo Nerium is intact, the Perinerium is intact. The only thing you're damaging is the Epinerium. Yes, the answer is C. Here's another question. Okay, so we're trying to do the patellar stretch reflex where you're like tapping the tendon. Well, patellar ligament, which stretches the tendon, the quadriceps tendon. The damage is in the central nervous system. So which of these things are not in the central nervous system? Which things are not in the central nervous system? Here's our sensory neuron. We can find the axon terminals of sensory neurons in the central nervous system. We can find the dendrites, cell body, and axon terminals of interneurons in the central nervous system. And we can find dendrites and cell bodies of motor neurons in the central nervous system. So which one of these is not in the central nervous system? Cell body is of the motor neuron, and dendrites are of the motor neuron. Axon terminal is of the sensory neuron, not the cell body. Can someone tell me where you'd find the cell body of a sensory neuron? In what is a term for the region where you find the cell body of a sensory neuron? I'll look in the chat. The answer is A. Where do you find a cell body of a sensory neuron? What is the term for the region of it, the location? Anyone get it? Anyone get it? It is in the peripheral nervous system, more specific than that. More specific than peripheral. What is the term for the cluster of cell bodies in the peripheral nervous system? What is the term for that? Gray matter is only in the central nervous system. So gray matter is only in the brain and spinal cord. What do we call a cluster of cell bodies in the peripheral nervous system? Cortex is also gray matter in the central nervous system. Still waiting. I know you can get it. Ganglia, yeah, there we go. Good job, Kelsey. Ganglia. Ganglia or ganglion. So singular is ganglion. Plural is ganglia. Okay. So I'm going to show you more histology and any other relevant models. I have to figure out if there are any more that I need to show you. And then I'll explain what the lab check and activity is. So yeah, let's look at this slide. This slide is called Spinal Cord Motor Neuron Smear. So basically, if you can imagine taking a spinal cord and just smearing it on the slide like butter on toast. Yeah. I am at 40x right now. We've stained for neurons, and this is actually a great shot right here. You can see a ton of neurons here. Neuron, neuron, neuron. Lots of different neurons in this area. You can see how big they are and how branched they are. You can see extensions at these different parts. Let me move around a little bit more. The reason why we have to smear it is so that we can see all these little extensions. Because if it weren't smeared, we wouldn't be able to see all the different parts. Okay, here's a huge cluster of neurons and other extensions. What I want you to notice about this, yes, here's the neuron and its nucleus is going to be in here, but we see more dots throughout, spread throughout. Those are glial cells. These dots are nuclei of glial cells all around. These are all glial cells, all those dots of different kinds. There's only one one glial cell where I can say with more certainty what kind of glial cell it is. If it follows along this long protrusion, it's likely one type of glial cell. First, can someone tell me what this extension of the neuron is called? What do you call this really long, broad extension? Yeah, it's the axon. And if we have motor neurons, we see cell bodies going all along the motor neuron axon. What kind of glial cell would we find here? What kind of glial cell would we find along the axon? That's right, Schwann cells. Because these are motor neurons, their axons extend into the peripheral nervous system. Those would be Schwann cells that are forming it. So I'm at 40x right now. I'm going to go to 100x. I'm searching for a neuron where I can easily point stuff out. So bear with me. Suppose this will do. Now, let me find one more. Huh. Okay, let's go with this one. All right. So it's stained very dark. And I should probably go to that one because it's stained less dark so we can see more things. So these neurons are branched, they're dark. Yes, I'm tired. How do we tell the difference between axons and dendrites? Think of dendrites as extensions of the cell body. In the cell body, yes, you have the nucleus, but you have all these organelles. And so these organelles are going to be stained a color. So that color extends into the dendrites. If you see that color extending into a branch, those are dendrites. But when it starts to get lighter and you have a really long projection, that would be an axon. So this one here, it's darker on these extensions, but this extension is lighter. This is likely the axon, this one here. So look for the lighter color. The lighter color is the axon. Dendrites have more color because organelles and stuff can get in there. The term for that is a Nissl body. I'm not going to test you on that, but in case you see that term, Nissl, it's just talking about the dark stain that you see within everything. Do axons connect to dendrites or directly to the cell body? Depends. If it is if it's a pseudo unipolar neuron, it goes straight from dendrites to axon. But if it's a bipolar or if it's a multipolar, it goes in order from dendrites to cell body to axon. Hope that answers your question, Kelsey. Okay, so those are neurons from the spinal cord that have been smeared so we can see individual parts. Let's take a look at an actual spinal cord that hasn't been smeared. So you can see there are 4 pieces of spinal cord. They're from 4 different sections. They're from a cervical section, thoracic, lumbar, and sacral. Cervical is this one. I don't know, it's kind of mirrored here. Cervical is over here. Thoracic is the small one in the middle. Lumbar is bigger. Sacral. The reason why it goes big, small, does anyone have a guess? Why does it go bigger in the cervical area, smaller in the thoracic, bigger in the lumbar, smaller in the sacral? Go ahead and type your guess in the chat. Spinal curve, it doesn't quite have to do with the curve, no. Think about what you find in the cervical location and the lumbar location that you don't have in the thoracic or sacral. It's not so much the curve. Well, why would there be bigger foramina, Isabel? Foramen means a hole. What are you trying to fit in there? Why is there more nerve? Why is there more spinal cord? What is in the cervical and thoracic areas? More room for spinal nerves that go to and from where? You're on the right track. More room for spinal nerves that go to and from where? Anybody? Help her out. Different regions, brain to the appendages. There we go. Yeah. To the appendages. Great job. It's bigger in the cervical area because it's going for your arms, bigger in the lumbar area because it's going to your legs. Your thoracic area, yeah, you got a big thorax, but it doesn't have as much innervation because there's just not much to send information to or from. So that's why the cervical and the thoracic ones are bigger. But regardless of which area we're looking at, they have the same basic pattern. They all look like this. Note that this is where the vertebra is. So this is the posterior or dorsal side. We're going to use dorsal ventral a lot. This is the posterior or dorsal side. This is the anterior ventral side. There's a larger, it's called a fissure. There's a larger fissure here on the anterior side. So we're looking at a spinal cord. This is the very center of the spinal cord. It's called the central canal. I'm going to go magnify on this. We're at 100x plus some more. What cell type is this? It's lining the central canal. What cell type is that? What cell type do we find lining the central canal? What is the cell type? Anyone? It's not simply cuboidal. It is a type of glial cell. It is a type of glial cell lining the central canal. What is the glial cell? What is its name? Osteoclasts only found in bone, elastics found in connective tissue. We're in nervous tissue right now. This is nervous tissue. So it's going to be a neuron or a glial cell. It's not a neuron. It is a glial cell. What is the name of the glial cell? What do we find lining a cerebrospinal fluid filled space? It's not satellite. Not satellite. It's lining a space, lining where the cerebrospinal fluid is. Yeah, Isabel, you lied. Good job. Ependymal cells, yeah. These are ependymal cells lining the central canal. Whether we're in the brain, which I'll show you coming up, or in the spinal cord, if it's lining a space, a fluid-filled space, those are ependymal cells. Remember what I said about gray matter in the spinal cord? Gray matter is in the horns. So horn, dorsal horn, ventral horn, dorsal horn, ventral horn. It's in these horns. So in our histology, the horns are right next to this central canal. There's a neuron. You can see its extensions, but its other projections with dendrites and axons have been cut off. That's why you just see this much of it. There's a neuron, 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 neuron. You see a bunch of other cells. They're smaller. Those are glial cells. Which glial cell are they? Well, we have 4 to choose from because we're in the central nervous system. The 4 to choose from are oligodendrocytes, astrocytes, ependymal cells, microglia. They're not ependymal cells because ependymal cells have to be lining a space. So that leaves us with microglia, astrocytes, what did I just say? Oligodendrocytes. What most of these likely are astrocytes. You can't tell. We need a special stain for that. And that's why we have a different slide. I'll go to that different slide in a second. So this is the gray matter. This is gray matter of the spinal cord. Going back to 40X, central canal. This is the gray matter. You can see the nuclei of the neurons here. If you just go down a little bit, we're here, and we're going to go down to here. To go down a little bit to this white column, this anterior column over here. We're at 100x total. What you can start to see are axons surrounded by myelin. Axons surrounded by myelin. Axons surrounded by myelin. These are mostly axons. It's not like the peripheral nerve. Only with peripheral nerves in the peripheral nervous system do you have those connective tissues. You don't have that same kind of connective tissue here in the central nervous system. Are all glial cells found in white matter too? Good question. When you're in white matter, you'd mostly find, you can find other glial cells, but you'll find more oligodendrocytes here. So you'll mostly find astrocytes in the gray matter. You'll mostly find in the central nervous system oligodendrocytes in the white matter, because they're the ones creating the myelin for these axons. So again, here's that border. This is gray matter. This is white matter. I know this is darker. It's just because of the stain. So this from here over is white matter. This way is gray matter. We know this because we see the large nuclei of, there we go, the large nuclei of the neurons. Give me a second, just trying to find the right spot. Where are you? There you are. Okay, I'm at 40x. We're at a new slide here. The slide is entitled Dorsal Root Ganglion. What is the dorsal root ganglion? Back to our spinal cord vertebra model. Here's the spinal cord. You have these extensions of a neuron. These are axons coming in. Like, these are afferent pathways. And here's the dorsal root ganglion. See how it comes in on the dorsal side? This is the dorsal side of the spinal cord. So that's why it's the dorsal root. It's coming out like a root, like roots off of a plant. So this is the dorsal root, and here's the ganglion for it. Ganglion, again, means collection of cell bodies. Can someone tell me, if this is an afferent pathway, you've got neurons, sensory neurons here in the dorsal root ganglion, what is the shape of the neuron? Is it multipolar, pseudo-unipolar, or bipolar? The shape of the neuron found in these dorsal root ganglia. Shape of the neuron. Lag is killing me. Anyone? Yes, great. Pseudo-unipolar. There are pseudo-unipolar neurons in here. Remember what that means. Pseudo-unipolar means the cell body is off to the side. Pseudo-unipolar neurons, these sensory neurons, the cell body is off to the side, the axons are running in a different way. So when we look at this is what we're looking at. We're looking at this dorsal root ganglion in histology. We have a bunch of these cell bodies clustered in one area. But then in other areas, we have very few cell bodies, very few neuron cell bodies. All of this is axon. These are all axons in longitudinal cut. These are axons that have been longitudinally cut. These are neuron cell bodies clustered together off to the side. neuron cell bodies off to the side, longitudinal cut of axons. Let me go to 100x. Okay, we can see everything here in one shot. So this is the neuron cell body, neuron cell body, neuron cell body. They're larger than the rest of these cell bodies. So the rest of these are glial cells. Whenever you see these little dots, those are nuclei of glial cells. We'll get to what they are in a minute. So here's the cell body of a pseudo unipolar neuron. Its cell body's off to the side. Its axons are running along here. We have a longitudinal cut here. Since we have what do you call it? We have nuclei running along these axons. Can someone tell me what kind of glial cell is this running along the axon? What kind of glial cell is running along the axon of this pseudo unipolar sensory neuron? If you answered Schwann cells, you are right. These are Schwann cells. We can assume that these are Schwann cells because we're in the peripheral nervous system and they're running along the axons. These would be Schwann cells. There's one more glial cell type that we can see here. Again, here's the neuron, and we see that these glial cells aren't over here by the axons, they're surrounding the neuron. You could say that they're orbiting the neuron. Can anyone tell me what glial cell is here? Surrounding these neurons? I need a timestamp. Does this work? Can all hear me okay? These are satellite cells. These are satellite cells. So satellite cells are surrounding the neurons. Glial cells are over here forming the myelin of the axons. Satellite cells, glial cells. This is from a dorsal root ganglion. Okay, this slide is from the brain. Where in the brain are we looking? Here's a longitudinal cut. Sorry, not longitudinal, sagittal. This is a mid-sagittal cut of the brain. You can see these spaces here. Those are called ventricles. Fluid-filled spaces are called ventricles. And they're all connected. The ventricles of the brain connect to this canal, which then continues down to the spinal cord, and that's how we form the central canal. All that cerebrospinal fluid is connected. So in this one particular part of the ventricles, labeled here in pink, Kelsey, satellite cells surround the body of dorsal root ganglion neurons. Yes, you're right. Kelsey, you're right. This thing within the ventricles is called choroid plexus. Choroid plexus. Choroid plexus is what you see here. And again, you find it in ventricles of the brain, those spaces of the brain. Choroid plexus is lined with the ependymal cells. Ependymal cells are the ones producing cerebrospinal fluid. This is where cerebrospinal fluid is being made. These cells that are lining in this space. You see how much surface area there is so they can keep generating fluid. They're surrounding blood vessels to take nutrients and water out. This is 40x. Let me go to 100x. These are ependymal cells of the choroid plexus. They're producing cerebrospinal fluid. So we're in a ventricle of the brain right now. You only find choroid plexus in the brain. You don't find it in the spinal cord. Choroid plexus. Bear with me. Okay. So this is from the brain. We don't see a whole lot right now. You see all these little specks. This is a special stain of brain tissue. So we're in the central nervous system. I'm at 40x right now. Let's go to 100x. Total. Right. Okay, what this is stained for? This has been specially stained for astrocytes. Here's a great example. Small cell body, very branched in all directions. Astrocyte, 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 and so on. This has been specially stained for astrocytes, not for neurons. Neurons would be much bigger. The cell body of a neuron would be bigger. Let's see if I can find an outline of one. Usually you can see it lightly stained. You know what? You can actually see outlines of, this has been specially stained for astrocytes, but you can see the faint outline of a cell body. See this lighter, darker thing? That's a cell body of a neuron. So it's like at least quadruple the size. Astrocytes are small, very, very branched. They're for general supporting of cells, supporting of neurons, and they also help form that blood-brain barrier. Astrocytes. We're still going to stay in the brain, but we're going to go to that cortex. Remember, cortex means outer layer. Just so you can see what I'm seeing. Hold on. There we go. Okay, so this is the outside of the brain. It's like if we were looking at the outside of one of these layers. We're looking like in this gray matter here. This is gray matter. This is gray matter. White matter would be deeper in. See all the axons kind of curving and going in a direction? That's white matter. All the cell bodies of neurons are down here. You can see all these cell bodies here. So this is gray matter of the cerebral cortex. Gray matter of the cerebral cortex. When we look at gray matter in the cerebral cortex, we see a special shape of neuron. It is multipolar. It is found in the central nervous system. But this neuron has a specific name. Let's go closer so we can see its shape. So two great examples are this one. You can see the nucleus and its shape right here, dendrites extending out. Cell body, dendrites, axon. They're pretty triangular. The term for these cells is pyramidal neurons. They're pyramidal, there we go. Pyramidal cells in the cerebral cortex. The cortex of the cerebrum. When we look at our brain, the cerebrum is what you normally think of, this largely wrinkly outer part. This is cerebellum. We'll get to that in a second. Cerebellum is back here. This is all cerebrum. So cerebral cortex are the neurons. We're looking at neurons in this cerebral cortex. They have this pyramidal shape. So they're called pyramidal neurons, shaped like pyramids. Here is more cortex, but this isn't the cerebrum. Just like you have folds in your cerebrum, you also have folds in your cerebellum. You can kind of see that here. Got folds in your cerebellum. Your cerebellum is this region back in the back of your head, back of your brain. Its overall purpose is for balance, a sense of balance, like balance and coordination. When you balance and coordinate, that means you need to get sensory input about what it is you're seeing. Your vision helps your balance. What you hear helps you balance. What you feel helps you balance. So you're taking lots of sensory input, and then you're controlling something. So yes, these are multipolar neurons, but dendrites are especially important for this neuron type. Dendrites are especially important. Let me. You can see this is gray matter right here. This is gray matter. In here, that'll be the white matter, but this is where you're going to find the cell bodies. Specifically, these are the neurons. They're called Purkinje cells. Purkinje is a guy's name. Purkinje cells. I'm going to go back to, my computer signed off. Give me a second. I'm going to show you what it looks like if you have a really good stain. If you have a really good stain, it'll look like this. You can see the cell body right here, just this small thing. All of this, these are dendrites. Very, very, very prominent dendrites because it's receiving so much information from other places. Purkinje cell. With our staining, with our light microscope, we're not going to see this much branching, but there's still branching. So right now we're at 40x. Here's the cell body. You can see a piece of dendrite coming off here, but the dendrites are going to be all branched in this area. The dendrites are in that area. Going to 100x. You can see it a little better. Cell body of the neuron, dendrites that are extending out here. We're just not seeing the branching like before. All right, finishing at 2:15. What do I have more to go over? I just have a few more slides to go over, and then I'll discuss the lab check and activities. So we'll probably finish before then, but I'll be here if you have any questions. So once again, these are they are in the central nervous system, so they're multipolar shaped, but the term for these cells are Purkinje cells. They're found in the cerebellum. They're found in the cerebellum. I believe there's one more slide I wanted to show you, and that is a teased or medullated nerve. When you tease hair, that means you separate your hair strands, and so it gives it like a big frizzy look. So this is, if you take a nerve and tease it, you're spreading it apart. We're separating the axons from each other so that we can see individual axons. All right, here we are at 40x total. You can see individual axons. These are all individual axons. Let's go a little bit closer. Let's go to 100x. I'm going to try to find one that's a little bit separated from the rest. That's good enough. What you can see here, this is an axon. You can see it alternates dark, light, dark, light, dark, light, dark, light, dark, light, and so on. These aren't striations. Striations are stripes that you see in muscle fibers. This is not that. Can anyone tell me what these lighter things represent? Who can tell me what we find at these lighter intervals? What is there? Or what do we call that region, rather, where it's lighter? If you answered node of Ranvier, then you're right. In these gaps, these are the nodes of Ranvier. Those gaps, let me spell it out again. Nodes of Ranvier. That's what you have at the, that's what these gaps are called. So that's where you have high concentration of those voltage gated channels. You can see the actual axon, this thin strip in the middle. This is the axon node of Ranvier, and these would be Schwann cells at every interval covering up, because this is a peripheral nerve, so those would be Schwann cells forming the myelin sheaths at those regular intervals. Okay, that's all the histology. There was a lot. You have this recording, and I made recordings. If you check the playlist channel for histology, it's all there. If you have any questions, type them in now. I'm going to start explaining, what's it called? Activities in lab check. Okay. Here is what we're going to do. So this is the last part, and then I can stay and answer any questions or explain anything again. But the two things that are due next week, are lab check 8, and activity 7. Let's take a look at activity 7 first. To find activity 7, you're on the home page, go to assignments. Under assignments, go down under activities. We're doing activity 7, workout plan, and this should show up. Oops, sorry, I gotta keep this in front here. Okay, so once again, what I did was, I was at home page, I clicked on assignments on the sidebar, and then under assignments, I found activity 7, workout plan. What I'm asking you to do, you're a personal trainer, you're acting like you're a personal trainer, or maybe you want to be one, so that helps. You're a personal trainer for someone preparing for an upcoming competition. You need to come up with an exercise plan that strengthens the muscles for that particular competition. So I want you to create a set of exercises, a plan for what exercises they should do to prepare for that specific competition. What should you consider? I want you to cover at least 3 muscles, 3 major muscles that we've discussed. And the movements and actions that are done with those particular muscles. Choose at least 3 muscles to use in your plan, and the corresponding movements and actions with those muscles. So let's say I'm working on my biceps brachii. That means I'm going to be flexing at the humeral ulnar joint. Please use technical terminology just like we've always done. So if my competition is something that involves this motion, I would be trying to find an exercise where you're flexing the humeral ulnar joint to strengthen the biceps brachii. So choose 3 major muscles. Choose 3 exercises that work those muscles, at least 3. Often one exercise will get more than one muscle, so that's great. But try to choose 3 different exercises. So let's say I want to do curls and hammer curls and then pull-ups, but like chin-ups. Those are all same motions, but they're 3 different exercises and they're going to be getting different muscles. So explain when you explain your 3 exercises. Explain how to do each exercise. Explain how to do each exercise using anatomical terms. Just like what we've done with the dances, when you explain how to move certain ways, same type of phrasing here. Explain how to do each exercise. Explain which muscles are being trained. And provide pictures or make your own drawings. It's fine if you copy pictures from other websites, that's fine. Or make a drawing to show how to, demonstrating how to do that exercise. So that's what I'm looking for. In your plan, you're explaining the muscles that you're working, what they do, what exercise you're doing, and how to do them with pictures. I want to you know what? I'm going to open this up. I have 8 for you to choose from here if you can't think of a competition, but I'm open to other suggestions if you want to pick a different competition. There's a reason why I chose these, because each one of them focuses on a different muscle group. So whatever competition you choose, make sure it's a single motion. Like if you do weightlifting competitions, you're going to be doing many different things. Maybe if it were a specific event in a weightlifting competition, that could be it. But yeah, if you have a different idea, run it by me. But otherwise, here are 8 that you can choose from. How do I want you to submit it? It could be a Google Doc. You could draw it out on paper, then take a picture of it and submit the picture. Whatever's easier, as long as I can access it. Make sure if you're doing a Google Doc that you share it as anyone with the link can access it. Because if you share it to Acon at DVC, I have my own Google account and then I can't access it. So make sure it's shareable. But there is a submit button thing here. The other thing is I want this to be individual. This is not a group activity. This is individual. Each one of you will be choosing one of these or one of your own and making your own workout plan. This is not due today, it's due in a week. This is due in 1 week. I will, of course, be checking to see that you've done your own work. Use your own phrasing, things like that. So whether that's copying straight from another website or copying from other people, I can tell. So please don't do that. Any other questions on activity 7? You pick one competition, you pick 3 exercises to strengthen those muscles, Emilio. Is a muscle speed dating thing due today? Muscle speed dating, if you're in the Monday, Wednesday lab, it's due today. If you're in the Tuesday, Thursday lab, it's due tomorrow. Any other questions about coming up with a workout plan? I'm not expecting you to do like how many reps, how many sets, how long you should. I'm not looking for that. I'm just looking for the exercises. Can you do a specific movement in CrossFit? For example, clean and jerk competition. Yeah. If you're saying clean and jerk competition, then what are exercises you can do to prepare for a clean and jerk? Because you can't just go straight into cleaning and jerking. That's a very powerful motion. What can you do to strengthen the muscles that'll help you with the clean and jerk? Yes, you can do that. Any other questions relating to activity 7? Overhead press, that's an exercise. I need a competition. So I guess, is overhead press a competition for how much you can do? If you can come up with three other exercises besides an overhead press that work the muscles to help you with an overhead press, then yes. I hope that made sense. You come up with 3 other exercises to help you prepare for that competition, go for it. Can you get extra if you send a video? I'm not opposed to that. If you, let me think about this real quick. What would I want in the video? If you, yeah, sure, okay. 3 points extra credit if you do a video, not just doing the exercise, but explaining it just like how I'm explaining all this to you. 3 points extra credit if you walk through it step by step. If you're doing workout for this competition, here are 3 exercises you can do. To do this exercise called a bench press, you have to take a bar, start in this position, extend at the humeral joint, and all that stuff. You're working these muscles. If you get information included in kind of like your major findings, I suppose. Maybe come up with a script before you record it, send it by me, and then I'll let you know. Send a script or an outline, and I can let you know if we're on the right track. Okay, that's activity 7. If you have more questions on that, let me know. Let's talk about the lab check. Honestly, I haven't decided what I want to do, so let's try to figure this out. Again, how to find lab check? Go to, from the home page, click on assignments. Under assignments, go down to syllabus and lab exercise checks. It's lab check 8 we're on now, on lab check 8. So here's lab check 8. We're gonna, since you're used to the having different parts, we'll just keep it at each person sticks to a part. So communicate with your lab group members. You each take a part. That's what you're responsible for. That's what you're graded on. So for example, person A is going to be looking at histology of different things, answering different questions from the lab book, and then giving an explanation of the other parts. So I still want those explanations of other parts. So be in communication with other people in your lab group. And even if they don't hold up their end, I still want you to talk about the other parts. So brush up on what, you should be brushing up on what they're doing anyways. So look at what it is that they're responsible for and the type of things that you need to know. So I still want explanations. You can look up stuff in the lab book. The lab book is all digital. It's the digital copy is online. So if you need to go to page 253, question 8, you can find that. Here to ask you to redraw it. You can either redraw it, take a picture of it, and upload that, or if you know how to digitally do that, like if you know how to edit a PDF, by all means, go for that. Here's the big change. Since we're not in lab, I'll get to you in a second, Isabel. Since we're not in lab for to look at histology, to look at microscopes, what I need from you to do is to find unlabeled images. I need you to find unlabeled images of the particular histology. Download or screenshot that and then label it. So what I want is, let me show you an example. Okay, here's an ideal way to do it. Let's say you open up a Google Slides document. Okay, here's my Google Slides document. Add a blank slide. This is my lab check 8 histology. And then my first thing, what was the first thing for this person? Histology of a neuron. So neuron, cell body, nucleus, dendrites, and axon. Okay. What that means is, what I want is, for you, I'm basically wanting you to sketch, remember that motor neuron that we saw smeared from a skeletal muscle, or skeletal muscle, from a spinal cord? That's what I want. So we need to find that somewhere. Where I would recommend you to look for that, here's the first place I would say you should go to. Go to Histology Guide. histologyguide.com. I really like this website because it's going to give you unlabeled, and then you can label it yourself. Unlabeled pictures of histology, and they're really good pictures. So how do you navigate this? You could use the search. Could use the search tool, or you could use the slide box. I'm going to use the search tool. We're trying to find a neuron, so let's give that a shot. So under search, I'm going to type neuron. Let's try this one, nuclear morphology motor neuron. Let's hope it has the right thing. It might not. I could look silly in a second. Give me a second. Okay. This is kind of what I want, but also not what I want. So what's shown here is a neuron that's in. This is showing neurons in within the tissue. I want a smear of neurons. So this actually won't do. This would be great if you're showing gray matter, because here we see neurons in gray matter. If this doesn't work out, what you can do is go to my Google Photos. So how do you get to that? Remember, go to Home. Go to Home and then scroll down, click on Shared Photo Album. This photo album is organized in order of what we go over. So we started with fetal pig. We did the banana activity, cells, mitosis, connective and epithelial tissues, skin, bone and cartilage, bone, lots of bone, muscle, lots of muscle, lots of muscle, lots of muscle, more muscle, okay. And dance, okay, great. Kind of like this, how we have two different, here's a neuron smear, unlabeled. Then here's a neuron smear labeled. I want both. I want unlabeled and labeled. So what you can do is you can take the image and copy it or screenshot it, whatever you need to do. So here I'm copying it. Go ahead and paste your image. So here's the unlabeled version. I'm going to copy this slide. So now I have two copies of the same exact thing. And I'm going to label this one. So this is unlabeled. This is labeled. I'm going to label it by selecting things like at the top menu, you can add an arrow. So here's the arrow tool. I'm going to change the color just so you can see it and make it thicker. So I can change the line thickness. I'm going to change it like 4 pixels. Where's color? In color, green, that stands out. So now I can point to different things. Whoops. Can point to the cell body, point to the dendrites, point to the axon hillock, and then I can type. I can add text. So if you're using a desktop, I feel like this is a simple way to label it. If you're proficient in Snapchat or Facebook Messenger labeling or any other photo program where you can draw directly onto a photo, then that's also good. I'm fine with you drawing arrows and typing in labels. So this is what I'm looking for. One slide unlabeled, one slide labeled. So it's just the exact same thing with and without labels. Oh, sorry, one more thing, what I would like you to do. On the unlabeled one, I still want those arrows. So I guess the simple way to do this is first make your labeled version. Then to make your unlabeled version, just delete the labels. Did that make sense for histology? Labeled and unlabeled versions. Questions? I'll wait for questions. Isabel or anyone else who have fewer than 4 people in your group, pick whatever letter you want, and that's what you're responsible for. If you want to continue working on the others, because it is good practice, go for it. Sorry you won't earn extra points. I still want you to talk about the other parts. We are supposed to form new groups next week. We'll see what happens, figuring things out as we go here. Okay. So the lab checks, this lab check number 8, is submitted online. It'd be nice if it were all in one document, like a Google Slides or something, or a Word document, something. It just makes it easier for me if you did it all in one document. You can if you write it out on paper and take pictures and upload that, that's fine too. Just as long as it's all there, just like before, as long as you upload everything, we're good. Let me make sure. Keep asking questions if you have any. Let me make sure there's anything else. If there's anything else I need to cover. Today is 16th. We're going over nervous tissue. Lab check 7 is due today. Next time, I will show you a live, well, not live, but an actual brain. We'll be going over brain models, as well as real human brains that came from our cadavers. So to recap, I know there are still issues with this and with the mini quiz. I'm probably going to drop this mini quiz score because it's too many people had issues with it. As I figure this out, I probably have to drop that score, just so you know. So if you weren't able to do it, don't worry about it. It's not going to count against you because I'm still figuring this out too. So we'll figure out what to do about this. I'll keep trying. If you haven't submitted your homework yet, lab check 7, your packet. If you haven't submitted it yet, please submit that now. We've finished chapter 14. Activity 7 is due, if you're Monday, Wednesday lab, it's due Monday. If you're Tuesday, Thursday lab, it's due Tuesday. So whatever your lab is. Activity 7 is the workout plan. Lab check 8, that's, you're doing the same things before, picking your part and going with it. But the new format is that for histology, what I'd prefer is if you created a set of slides, one with leader lines, the other with lines plus labels. So basically one's labeled, one's not, but you should add leader lines. It should not already be labeled. Thanks, Saldi. It should not already, oh no, it's Corey, damn it. It should not already be labeled. And yeah, don't forget about your, thanks, Corey. Don't forget about your discussion. Which one is it? Chapter, whatever. Don't forget about your discussion. I think that's it. I'm not going to end I'll stay live for a bit longer. I'm going to wait for questions. If you want me to go over anything that I've already gone over, if you want to see it again, I can do that too. But activity 7, do Monday or Tuesday. Lab check 8, also do Monday or Tuesday. And they're both individual-ish, but you're working together for that explanation. We're trying to get info from your lab mates, your virtual lab mates. Huh? Anything? No? If you have suggestions for intro and outro music, let me know. Anyone's a good sound or video editor? I mean, I can do it. I just don't have the time. But yeah, so if you're, Corey, I see your question. Or yeah, Corey, I see your question. Corey, do you mean histology or just them in general? But anyone else, stick around if you want. But I'm just gonna review stuff for whatever questions you have. Sure. Okay. Let's put this back over here. Turn the lights down a little bit. Okay, we talked about glial cells on slide. We started on slide 14. You're welcome, Najas. Slide 19 or 14. Slide 14. When we look at histology, neurons are typically very large, very big and branched. You see nuclei surrounding them, those are some kind of glial cell. So it's about narrowing down which glial cell are we looking at, because there's a lot of different kinds and they do different things. If we're in the peripheral nervous system, we have two choices. In the peripheral nervous system, we have satellite cells and we have Schwann cells. Satellite cells are the ones that are immediately surrounding the cell bodies. So we saw this in the dorsal root ganglia. Let me find it for you. We are looking at, oh, hold on. All right. I'm at 40x right now, going to 100x. Great. So here we have neuron cell body, neuron cell body, neuron cell body, these larger ones. Surrounding them, those are satellite cells, and they're there to just help this neuron function properly. Over here, these are all axons, but we see cell bodies. We see nuclei here. So what nuclei would be bordering axons? Those are Schwann cells. So Schwann cells form myelin sheaths. Satellite cells are just generally supporting the cell bodies of these peripheral nervous system neurons. If we're in the central nervous system, one of the more straightforward ones to find, if you see one lining a space, whether it's a ventricle or a central canal, then that's an ependymal cell. Ependymal cells line compartments of ventricles, of central canal, and they're for circulating cerebrospinal fluid, producing and circulating cerebrospinal fluid. So when we look at that under the scope, here we have choroid plexus within a ventricle. So the white space is ventricle, where the CSF would be, and this is ependymal cells. These are ependymal cells. We're at 40x right now, going to 100x. These are ependymal cells. I know they look like simple cuboidal. Someone said that, but that's what they are. Just like we have satellite cells surrounding cell bodies in peripheral nervous system, astrocytes are your major helper in the central nervous system. Star-shaped glial cells that you can better see if you have the right stain. So if we don't have the proper stain, here we are looking at a spinal cord cross section. You can see the central canal there in the middle. Central canal, all these darker stained ones, those are neurons. But then you see these lighter, smaller spots, those are glial cells. They are likely astrocytes, but you can't see that kind of detail here. So with our special stain, we can see it. So this is from the brain, spinal cord, it shouldn't matter, it's central nervous system. And with this special stain, we can go to 100x and more clearly see the star-shaped pattern of these astrocytes. So they're generally helpful, and they can extend their processes to form the blood-brain barrier. Microglia, we don't really have histology of that. So hint, I'm not going to test you on the histology of microglia, but you should know what they are. Just like we talked about Langerhans cells in general. Langerhans cells are for cleaning up things in the skin. Microglia are for cleaning things up in your nervous system. You don't have a direct blood supply. You don't have your neurons aren't surrounded by blood. They're protected because the astrocytes are wrapping around the blood vessels. So you need immune cells with it that are just dedicated for the nervous system. That's what microglia are. Oligodendrocytes. These are Schwann cells are for the peripheral nervous system. Oligodendrocytes are for the central nervous system, and so they're the ones forming the myelin. To find them on our slides, if I go back to that, spinal cord. All right, we are at 100x right now. So we get our bearings here. There's the central canal. Central canal, gray matter, you can see the nuclei of neurons. This is white matter. So this would be an axon. Can you see that? Like there's an example of an axon. There's an example of myelin. An oligodendrocyte might be one of these darker dots. I can't say for sure, but they would be in here somewhere. It's hard to find the histology of that too. So in the central nervous system, you've got oligodendrocytes, astrocytes, ependymal cells, and microglia. In the peripheral nervous system, you have satellite cells and Schwann cells. Any other questions, things you want me to go over? Review pictures I've drawn on the board. Of course. I'm going to put some models away. If you have more questions, I'll check in a minute. Just type it in the chat. Yeah, I can go over that. Okay. So debatable, but broadly, there are three shapes that neurons can have. Neurons can be bipolar, neurons can be pseudo-unipolar, and neurons can be multipolar. Bipolar and pseudo-unipolar, they're for sensory things. Bipolar specifically is for special senses, things like vision or smell. Those are examples of special senses. They tend to be shorter. Bipolar just means you got dendrites, cell body, axon, axon terminal, all in one line. And the dendrites aren't very extensive because they're just for sensing the one thing. Right, so bipolar cells, typically for special senses. Pseudo-unipolar neurons are for somatosensation. That's touch, temperature, pain, pressure, stuff like that. Pseudo-unipolar, you've got the cell body off to the side. You've got the cell body off to the side, and dendrites straight to the axon terminal. So pseudo-unipolar, we saw the cell bodies in part of the axon. We saw that in the dorsal root ganglion slide. That's what we were looking at. We're looking at this. Cell bodies all in one area, axons in another. Axon terminal would be farther along. So both are sensory. Special senses, somatosensation. Multipolar neurons could be in the brain, could be in the spinal cord. So they could be interneurons or they could be motor neurons. Multipolar because they have many dendrites, not just a few like these. They have a lot of dendrites surrounding the cell body. And usually just one major axon. This axon can branch. So this is just a simplification, but you could branch some more. But it's usually just one major axon which then branches to smaller things. Multipolar neurons, again, are interneurons, so either fully within the brain or spinal cord, or they're motor neurons, like somatic motor neurons are multipolar. If you want to take a picture of this, here's this drawing. If you wanted this one, what I was showing here, sensory afferent, interneuron in the central nervous system, motor neuron out here, efferent pathway. So then over here was just general anatomy of a neuron. Dendrites, cell body, axon hillock, myelin sheaths, nodes of Ranvier, axon terminal. Synaptic cleft, postsynapse. This whole thing is the synapse where one neuron's communicating to something else. And then up here, I was going over graded versus action potentials. A graded potential, a graded potential is where you have gradations of charge coming in. So right where sodium comes in, it's going to be very charged. But as it spreads, you're going to get weaker and weaker charge. So that's why it's a graded potential. If that graded potential is strong enough, if enough sodium came in that you get enough charge getting to the axon hillock, then you can activate an action potential. So going down the axon, starting at the axon hillock, that's the action potential. And that's activated by these voltage-gated channels. These ones here, these are chemically gated channels. Over here, we have voltage-gated ones. Any other questions? I'm going to look out for questions. I'm not going to end the feed yet, but if you do have questions, type it there and I'll get back to you in a minute. Okay, if anyone else wants to talk more, I can do that. Send me a message. But I'm going to end the live stream. Thanks. Crazy first day. Hope that helped. See you later.